Okay, sorry. Thank you very much for having me here, and thank you for your introduction. Well, I'm going to talk about mapping asymmetries in the U-shaped fiber system of the human brain. So very quickly for a historical overview of this topic. So as you may all well know, in the human brain, the association fibers can be classified as long and short association fibers. And this was first proposed by Theodor Hermann Maynard in the 19th century. And he was also the one to describe for the first time that in the superficial white matter, the axons close to the cortex form U-shaped bundles that connect adjacent gyri or neighboring gyri. Now, although this description has been there for a long time, most of the U-shaped fibers in the human brain have remained unmapped or uncharacterized. Now, with the recent advent of diffusion MRI and tractography, there has been a handful of studies that have tried to show these fibers with MRI. Now, these studies can be classified largely into two groups. So the studies that have tried to do a manual approach or virtual in vivo dissections in specific regions of the brain to show these tracks, such as the frontal lobe, or other studies that have attempted to use advanced clustering methods to distinguish between the large association tracks and the short U-shaped fibers. So the aim of our study here was to generate a whole brain probabilistic atlas of the U-shaped bundles, and also to map interhemispheric differences in these tracks. And this point is really important because if there's something that is constant across the vertebrate species is lateralization of function and structure. So in our study, we used 133 Human Connectome Project participants, all male and right-handed. Right-handedness was defined with a score of greater than 50 in the Edinburgh Handedness Questionnaire. We decided to use pre-processed diffusion and structural data. The diffusion was single shell with p-values of 2,000 seconds per millimeter square with 90 directions and 18 BCO volumes and an isotropic voxel size of 1.25 millimeters. Afterwards, the tractography was processed with Star Track, which uses a spherical deconvolution algorithm with a damped Richardson-Lucy. The spherical deconvolution parameters were as follows: an alpha of 1.5 with 400 iterations, and tracking was performed with standard tracking parameters except an angle threshold of 60 because of the highly curvature of these tracts. So once we have the tractograms for each of our subjects, we're interested in only keeping the U-shaped fibers. So in order to do this, we applied two filtering criteria. So the first one is a geometrical constraint, and the way it works is as follows. So say we have our U-shaped streamlines, like that one, and we have the endpoints. So if the distance between the endpoints of the streamline is less than the length of the streamline divided by pi, we keep those streamlines, and the other ones are discarded. So effectively, we're only keeping the streamlines that have a semi-ellipse. And this was presented by Raphael O'Halloran in ISMR in 2016. The second constraint is a length threshold between 20 and 60 millimeters. And this arbitrary threshold was ascertained by what the literature says that most of the U-shaped fibers have as length. And once we apply these filtering criteria, we obtain two things. One is the U-shaped filtered tractograms for each of the subjects, and subsequently its density map. So for our atlas generation, what we did was we used the density maps of each of the subjects. They were non-linearly registered to the M952 asymmetric template, and then later on they were binarized and averaged. For the asymmetry analysis, what we did was, using these density maps as well, we created a subsequent copy for each of the subjects, and we left-right flipped in order to compare the right hemisphere with the left hemisphere and vice versa. So we have now two data sets that were non-linearly registered to a symmetric template because we wanted to avoid biases due to gyrification. And we compare them using FSL's randomized, which uses non-parametric permutation testing, and a pair t-test. So the first result that I want to show you is this one, which is a concatenated tractogram of the 133 subjects. So it's basically combining the streamlines of all the subjects. And we wanted to do this so we could qualitatively check that our method was working. And as you can see in this image, it is quite striking that we can see a consistent pattern of U-shaped fibers connecting the adjacent gyri. Now, what you're seeing in the image as a scalar is the streamline density, and you can notice that there's some U-shaped fibers that have a higher density than others. And we suspect that this is because there are some U-shaped fibers that are highly consistent across a cohort of subjects, while others might be more variable due to differences in gyrification. Now, the next result is our probabilistic atlas, which we could observe that tracts that had been previously described in the literature, such as the frontal marginal tract that you can see in the frontal pole. This tract is underneath the marginal sulcus. We could also see the frontal insular tracts that are connecting the insular cortex with the frontal cortex. We could also notice frontoparietal tracts, which are very prominent underneath the central sulcus, connecting the precentral gyrus and the postcentral gyrus. And we could also see another tract that has been previously described as a frontocentral U-shaped fiber connecting the precentral gyrus with the middle frontal gyrus. Now, we could also observe other U-shaped fiber bundles that have not been previously described, such as a connection between the superior parietal lobule and the supramarginal gyrus underneath the interparietal sulcus, connections between the superior occipital gyrus and the posterior parietal gyrus, which are running underneath the superior occipital sulcus, and also ventrally, these ones connect with the middle occipital gyrus. And finally, another U-shaped fiber bundle that I'm going to show you is this one connecting the superior temporal gyrus with the middle temporal gyrus. For the asymmetry analysis, what you can see in the left-hand side panel is the p-value maps of our paired t-test. And as you can see, there's a striking pattern of asymmetry in which in the left hemisphere, we can see most of the significant voxels are in the cortex adjacent to the insula, the ventral frontal lobe, and the temporal regions, and as well as the lateral occipital cortex. While on the other hand, in the right hemisphere, we can see that most of the significant voxels were concentrated in the dorsal frontal lobe between the superior frontal gyrus and the middle frontal gyrus, and also in the temporal-parietal junction between the superior temporal gyrus and the angular gyrus. Now, we suspect that this is because of, and is in line with current theories that the left hemisphere has a specialization for language, while the right hemisphere is more special for cognitive execution and visual spatial attention. Now, in order to improve the visualization of these results, we used these significant voxels as regions of interest to see which of the streamlines were being captured in our large tractogram. And what you can see up here is the surface projections of the density map in that tractogram, which show the tracks, and we can see them more clearly in this little video, where we can see that in the left hemisphere, most of the tracks that are asymmetric are the ones in the insular cortex connecting with the frontal lobe, while in the right hemisphere, we're seeing a set of U-shaped bundles connecting the superior with a middle frontal gyrus. So finally, to conclude, we were able to present with a very easily and fast method a whole brain probabilistic atlas of the U-shaped fibers. We were also the first to show a comprehensive map of an interhemispheric difference in the U-shaped fiber system, showing that in the left hemisphere, there were higher local connectivity for language-relevant regions, while in the right hemisphere, we saw higher local connectivity for maybe functions as cognitive execution and visual spatial attention. Now, it will be very important in future work to see if these results hold using other diffusion and tractography models, because other studies have already shown that results can vary depending on the algorithm. Now, it will also be important to show or correlate these bundles with functional and behavioral measurements that are available in the HCP, because we want to know what is the cognitive role of these U-shaped bundles. Now, it's important to say that these bundles are the ones that are the latest to myelinate, so they myelinate even during the third and the fourth decade of life, which might indicate a very prominent role in cognition. And it's also important to notice that these bundles are often spared in some neurological and neurodegenerative diseases. And finally, the role of these tracks in potential patient populations such as autism will be important to explore. Thank you very much, and I would like to acknowledge my supervisors, the rest of the NAT Brain Lab, and my funding and sponsors. Thank you.
The floor is now open for questions.
Hey, Francisco, great talk.
Thank you.
Really nice work. I just wanted to ask, are there any adjacent gyri where you don't find a U-fiber connection?
Yeah, yes. Particularly in the occipital and the temporal lobe, we found some regions that did not have occipital, just short U-shaped fibers. Although we will have to explore if this is a limitation of the method or is actually no U-shaped fibers in those regions.
And do you think, do you also see the lack of those U-shaped fibers at an individual level? Or is it because the sulcal anatomy is more variable there or something?
Yeah, so I'm showing the results only at the group level, and I still have to explore if it's consistent across all the subjects. Thank you.
Symmetries are known to differ between genders. Have you been looking at that?
In this scenario, I only used male participants because I wanted to reduce the morphological variation in the brains. But currently, I'm repeating the same analysis using females and males. So, yeah.
Hi, thanks. That was great. Is it correct that you used some sort of spatial filtering on the data, if I understood it correctly?
It's not a spatial filtering, more than a geometrical constraint to limit, well, to only maintain the streamlines that have this geometry.
Okay, so the spatial observation scale was the same?
Yes,
in all
yes.
Okay.
That was a great talk. I was wondering, I saw a paper last year that all sports was on about polymicrogyria and the U fibers of the brain and how the cortex, like the structure, cortical thickness kind of folding can really affect U fiber architecture and even resolving U fibers in individual brains. And I was wondering if you can control for that any in like variations and structure, actual like cortical folding in the individuals, or if that was like not
if I remember correctly, there's a paper watching at U-shaped fibers from 2014 that have already seen correlations between gyrification and cortical thickness.